This
Can you hear
conference
me?
will now be recorded.
Yeah, yesterday we had finished replication of DNA. So we had spoken about how the genetic information will be transferred from the parental strands to the daughter strands, and we had seen that there was a leading strand and the lagging strand was seen, and how the replication occurs in the 5' direction to the 3' direction, both in the leading as well as in the lagging strand. Today we'll be talking about what is the DNA recombination and repair. So DNA recombination, it involves the genetic exchange or exchange of genetic material either between multiple chromosomes or between different regions of the same chromosomes. Now, why do the chromosomes they undergo recombination? What is the reason for that? This is because we said that exchange of genetic information between newly duplicated chromosomes is required during the process of meiosis. Then, to ensure that each gamete includes both maternally and paternally derived genetic information. Then, to promote genetic diversity, such that the resulting offspring will inherit genes from all of 4 of its grandparents, thereby acquiring a maximum amount of genetic diversity. Then recombination, other than this genetic inheritance or genetic diversity, it is also used in DNA repair, that is particularly in the repair of the double-stranded breaks. Then during DNA replication also, it is required to assist in filling the gaps and preventing the stalling of the replication fork. What happens if recombination does not take place? So deleterious or dangerous mutations may accumulate in each chromosomes. So if you remember the meiosis and meiotic division, this is a picture of a homologous pair, and you can see this is the centromere, and these are the sister chromatids, and this together they form the tetrad. So crossing over is taking place here. This is an example of crossing over that is taking place at this point, that is known as the chiasma. And then after the crossing over, this is the resultant chromosomes that are formed. This is only a picture. We will deal in detail what it is. Now, what is mitotic and meiotic recombination? So we said that recombination can occur both during mitosis as well as during meiosis. Now, only meiotic recombination, it serves the important role of reassorting the genes, while mitotic recombination is important for repair of mutation in one of a pair of sister chromatids. Then what are the types of recombination? So there are two mainly broad types. There may be either general recombination, or they may be site-specific recombination. Now, general recombination is again of two types. It may be homologous or it may be non-homologous. So we will be dealing mostly in detail about this homologous or non-homologous recombination. So what is homologous recombination is that these are the sequences that are nearly identical. Example, you can see during meiosis where there is an exchange between the maternal and paternal chromosome pairs, or non-homologous where the DNA element moves from one side to another. Usually, little sequence similarity will be seen when the DNA moves from one side to other. Example you can give is of transposition. Then site-specific recombination occurs between sequences with a very limited stretch of similarity, and it involves only the specific sites. So if you just take a simple example here, we have just given an alphabet. So here you can see these are alphabetical in the capital letters and in the small letters, but they are the same alphabets. So we are saying these are the given example as a parental chromosomes. So what happens in homologous recombination? What we said was the definition previously. sequences that are nearly identical. So exchange of information between sequences that are nearly identical will take place. So here, from here, the sequence from this chromosome will be shifted to this chromosome. So you can see they are having identical sequences again. So from both the in homologous recombination. So a recombination is occurring between similar molecules and then between similar sequences. So again, this is the same example like what we have seen. Then for homologous recombination, there are three models: Holiday model, Michelson model, and double-stranded break model. But we will be talking mainly about the Holiday model because this is the most commonest model that is being utilized. So this is here you can see these are this is a model that depicts the genetic recombination which can occur as a result of breakage and rejoining of the heterologous DNA strands. So we are talking here about the Holiday model here, and you can see these are the two pairs of strands of DNA over here. And in order for recombination to take place, an endonuclease will cause a cut in both the DNA strands. So here you can see this is a cut over here, and this is a cut that is made by the endonuclease. Then in the next step, there is a displacement of the strand. After the cut is made, displacement of strand is taking place from this point and from this point onwards. Okay. So first you have the two pairs of DNAs, and then from then followed by endonuclease that is cutting the strands. Then displacement of the strand is taking place in both the DNAs. Then afterward, the ligation of the strand. Here, ligation of the strands takes place. So this leads to the development of a heteroduplex DNA molecule. DNA molecules are formed over here. Once this ligation has taken place, then there will be migration of the branches. So branch migration will take place, and you can see this is the branch that is migrated from one DNA molecule to another, and this another that has been migrated. Now, even though this duplex strand we are showing over here, this will, when you want to present it as a Holiday model, this will undergo a 180-degree rotation to present this sort of a Holiday model. Now, this Holiday model, a structure that is showing in this form, it may undergo a nick. Either it may be a horizontal nick or it may be a vertical cleavage may occur in this strand. So it will give rise to different types of recombinant duplexes. So either it may form this type of a duplex, or it may form, if it undergoes a horizontal cut, it may form a two different, this type of a duplex can be formed. So this is an electron micrograph shows a DNA in a chi-form structure similar to the diagram that is seen over here. This is the Holiday structure, chi-form. So this is similarly it is shown in an electron micrograph. This is derived from E. coli. So if you just again, if we go through what is we have seen in the homologous recombination, we said these are the two pairs of DNA molecules. Nicking is occurring by the endonuclease. Single strand breaks are occurring. Crossing over of the strands is occurring, and then sieving or ligation of the cuts that are taking place. So crossing over of the branch migration takes place, resulting in the formation of a heteroduplex DNA. Then the rotation, 180-degree rotation takes place to form the Holiday junction. And then here, nick can be said, it can be a horizontal nick cut can take place. So you can see a horizontal cut, if it takes place, it can take place here, or a vertical cut can take place. So then the ends will be sealed by ligase. So here you can see this is the end result if it's a horizontal cut, or if it's a vertical cut, this is the recombination model that can occur of the DNA molecules. Then what do you mean by non-homologous recombination, which is also known as transposition? Now transposition, this involves a transposable element, TE, or it can also be called as transposon or retrotransposon. Another common name that is known as they are also known as jumping genes, because they are the mobile segments of the DNA sequence, and they can move from any place to any place in the target chromosome. So that's why they're called as jumping or mobile segments. They can move from one place to another place, any place in the chromosomes. And sometimes they may create mutation, or sometimes they may reverse mutation. And they alter the cell genome's size. So the recombination process occurs without any special homologous sequence of DNA over here. That means without any identical sequence. Homologous sequence means without any identical sequence of the DNA. And this transposition often results in duplication of the transposon element itself. So there are two classes of non-homologous recombination. So it may be either class 1, that is also known as retrotransposition, and it is also said to be a cut and paste mechanism which involves an RNA intermediate. While class 2 is a DNA transposition, this we say it is a copy and paste mechanism that is taking place over here. It does not involve an RNA intermediate, but involves the direct transposition of DNA to DNA itself. So we will see each one what it is. Now retrotransposition here, you can see this is the DNA molecule. This is the retrotransposon or transposable element or transposon, whatever you want to say. And what is happening here? This is undergo transcription to form an RNA. So this is an RNA that is formed through an RNA polymerase. An RNA is being formed. Then through reverse transcription by the enzyme reverse transcriptase, it will lead to formation of a copy of the DNA. So this RNA that we said retrotransposition is occurring through an RNA intermediate. So this RNA that is formed through transcription from the retrotransposon DNA sequence, retrotransposon nothing but a DNA sequence. So again, through reverse transcriptase, a copy of the DNA is forming. Now this DNA copy will get inserted into the target DNA. So here, this is the target DNA, and insertion of this DNA copy is occurring over here. So this is a retrotransposition. What we said retrotransposition is a cut and paste. So it is taking out from here and being pasted in a target DNA. So here again, this is another picture showing the retrotransposon only. So here you can see this is a retrotransposon which is having flanked by direct repeats, transcription to form the RNA, then reverse transcription to form the double-stranded copied DNA which get inserted. So insertion is taking place here to form the new copy of the retrotransposon. Then DNA transposition. This DNA transposition is of two types. You have replicative transposition, DNA transposition, and a conservative type of DNA transposition. So what is this replicative transposition? This replicative transposition, it requires single strand breaks, replication, and then resolution. So here you can see this is a transposable element, and this is the target sequence. This one in the purple is the target sequence. Now, a transposase is an enzyme which will bind with a transposable element. It makes a single-stranded breaks at each end of the transposable element. Here, you can see it is making single-stranded breaks over here. Then the free ends at each end of the, then the free ends of the transposable element will attach to free ends of the target sequence here. So the free ends, once the transpose makes the cuts over here, the free ends will bind to the free ends of the target sequence over here. Then replication will take place. Now these are single stranded. So replication has to take place on the single stranded templates beginning at the 3' hydroxy ends of the single strand. So here replication has taken place through the transposable elements in the target sequence. So you can see here the double strands are taking place. So what we are getting is that there is an integration of the A and B molecules, co-integration formation with 2 copies of the transposable element and 2 copies of the target sequences. Here it is taking place. This is only for explanation's sake, but this co-integrate resolution it will resolute by crossing over between the sites within the transposable element, and then 2 separate copies of the transposable element is taking place over here, but 1 new copy is flanked by direct repeats of the target sequence. So in the end, what is happening is that this is being copied over here to the new target sequence. Then we come to the conservative type of transposition. So here you can see this is a flanking direct repeats are generated when a transposable element inserts into the DNA. So this is a DNA, this is a target DNA with its sequence, direct DNA sequence, the target site of the DNA, and the DNA at the target site, it will be cut in such a way that it will produce sort of a sticky ends. Sticky ends like you see in restriction endonuclease, the sticky ends will be seen over here. And then this is a transposable element to which the transpose enzyme will bind. This is a transposable element with inverted repeats on the sites. With inverted repeats on the site to which the transposes will bind. This transposase enzyme will insert the transposon anywhere in the genome, or sometimes at even at target sites. So mostly it can insert anywhere in the genome and sometimes at some particular target sites. So here it is the transposase inserting the transposable element at where the target site in the DNA has been cleaved to make sticky ends. Now these gaps will be filled up with DNA polymerase, and then always it will be ligase will fill the sealing will occur with the ligase. So now it will have the flanking direct repeats are formed on either sides of the transpose element. So this is again the same structure. I want to repeat again here. This is another figure. So we said there are 2 methods of transposition: retrotransposition, which requires an RNA intermediate, which is a cut and paste mechanism. So this is a just simplification of where we are showing that transposon here is cut and inserted at the DNA sequence over here at the target sequence. While DNA transposition is a 2 type, replicative and conservative. So it is a sort of a copy and paste mechanism where the transposon is being copied and pasted at the DNA sequence, targeted DNA sequence. Then site-specific recombination. It is used both by eukaryotes as well as prokaryotes. This is mainly to regulate the gene expression as well as to increase the organism's genetic repertoire. Then the DNA, once replication occurs, DNA is always subjected to several damages. The causes are quite several. So it may be occur, the DNA damage may occur as a result of errors during replication, or it may as a result of physical or chemical agents, or it may as a result of mutation. So we will just see in a little bit in detail what are the types of damages that can occur to the DNA. So types of damage may be as a result of oxidation of the bases, as a result of alkylation or methylation of the bases. It may occur as a result of hydrolysis of bases such as deamination, depurination, and depyrimidation, as a result of adduct formation, as a result of mismatch of the bases due to errors in DNA replication, as a result of ultraviolet B or ultraviolet A light. So this as a damage that occurs due to ultraviolet B may cause cross-linking and result in the formation of pyrimidine dimers. This is a direct damage. While the ultraviolet A light creates mostly free radicals causing indirect DNA damages. Then you have ionizing radiation such as gamma rays that may lead to irreparable DNA damage. Then temperature modification, elevation in the temperature may also lead to depurination, loss of purine bases from the DNA backbone, and it may also lead to single-stranded breaks. Then again, you have the chemicals such as aromatic hydrocarbon that can lead to DNA adducts. Now, so many types of DNA damage can occur. For simplification, we have a classification which is based on a single base alteration category. We can say single base alteration which may occur as a result of deamination, as a result of depurination, that we said hydrolysis, alkylation, base alkylation of methylation, insertion or deletion of nucleotides. Or there may be two base alteration may occur as a result of UV light. We said UVB light that may induce pyrimidine dimer may be formed as a result of UV radiation, or chain breaks may occur as a result of UVA, free radical formation, oxidation may occur, then as a result of ionizing radiation. Then cross linkages. Cross linkages may occur between the bases either in the same or it may occur as a result of opposite strand. Then cross linkage may also occur between the DNA and the protein molecule. Now we will see what do you mean by, we said this, we will see one by one what is happening. In the single base alteration, what we said either deamination may occur, depurination, base alkylation, insertion or deletion of nucleotides. So here we are showing an example of deamination here. This is cytosine, and here the amino group of cytosine is being removed. So once the amino group of cytosine or cytosine undergoes deamination, it will result in the formation of uracil. Similarly, you have adenine over here. The amino group of adenine, if it's removed, it will result in the formation of hypoxanthine. So what is happening is that with single base alteration, cytosine is being converted to uracil and adenine is being converted to hypoxanthine. Then if you go for depurination, so here you can see this is a DNA chain, purine base, it may be removed. Like guanine here, we get an example of guanine from a DNA chain, it may be removed completely. So that in the DNA chain, what will happen at the site? The chain will contain only the ribose and the phosphate group without the base. So it is called as a AP site, apurinic site. If a pyrimidine is removed, it is called as a apyrimidic site. So it is an AP site. So without having the purine or pyrimidine. So depurination or depyrimidation may occur here over here with the loss of either of the bases. Then alkylation is nothing but methylation. Here you can see this is the structure of guanine. This guanine, if it becomes methylated or undergoes addition of an alkyl group is done, added to it, it forms methyl guanine. Methyl guanine is formed over here. So alkylation and alkyl group, methyl group get added to the bases. This may be chemical induced. Some may be harmless or some may cause mutation by mispairing during replication, or some may stop the polymerase altogether. Then the next is exposure to UV light, we said two base alteration that may occur in the result in the formation of thymine, this pyrimidine dimer. This thymine dimer is the one which is most commonest, or it may forming a thymine cytosine dimer may also occur. So it occurs as a result of UV radiation. B and sometimes C also. So here you can see that exposure to UV light, here we are saying this is a formation of a thymine dimer that is occurring as a result of UV radiation. So here together we can just see here what we said. A single base alteration that may occur as a result of deamination. Here, removal of amino group, forming of cytosine being converted to uracil, adding to hypoxanthine. Depurination where guanine can be removed and from the DNA chain resulting in an AP site, either apurinic site or apyrimidic site, or addition of a methyl group may occur resulting in the formation of methyl guanine. Then we said exposure to base alteration may occur with the formation of pyrimidine dimers. This pyrimidine dimers will contain the covalent bonds. Then other than that, we also said there may be damage that may also occur as a result of oxidation, formation of free radicals where guanine oxidizes to 8-oxoguanine, and also it may lead to single-stranded or double-stranded breaks, which are very important in the organelles. Then as a result of error in replication, a wrong nucleotide or a modified nucleotide may be inserted. Then double-stranded breaks may also be seen. This is induced by ionizing radiation as a result of transposons, as a result of topoisomerases, or as a result of homing endonucleases or mechanical stress on chromosomes. So double-stranded breaks, there are several causes are there, and then this double-stranded breaks will also repair can also occur with the breaks. So now how does this, we are saying so much DNA undergoing damage by so many mechanisms. So there are also several mechanisms to take the repair of the DNA. So we will be seeing what are the mechanisms that will help in repairing of the DNA damages. So these are the different types of DNA repair. They may be base excision repair, they may be nucleotide excision repair, mismatch repair, and double strand breaks repair. So 4 types of DNA repair are seen. What are they? Base excision, nucleotide excision repair, mismatch, and double stranded breaks repair. So what is this base excision repair? The bases cytosine, adenine, and guanine, they can undergo spontaneous depurination as we have just seen to form uracil, hypoxanthine, and xanthine. Now these altered bases, they do not exist in the normal DNA and therefore they have to be removed. So here we are showing in this figure how a base excision or a damage or a base that is being replaced. Yeah. So here is an example of cytosine which has been deaminated to uracil. So this U here represented the uracil that is being taking place instead of cytosine, that has occurred instead of cytosine over here. So here an enzyme DNA glycosylase, here it will remove the defective base uracil. So from DNA glycoside will remove the defective pair uracil. So this results in either apurinic or a apyrimidic site depending on whether purine base or a pyrimidine base is removed. So once this is what happened, then the next step is the action by AP endonucleases. So this AP endonuclease enzyme, they recognize the sites with the missing base and they cleave the sugar phosphate backbone. So this apurinic site will contain only the sugar and the phosphate that's remaining behind. So this endonuclease will cleave the sugar phosphate backbone and this removes the sugar and phosphate that's lacking the base. So this deoxyribose phosphodiesterase removes the sugar phosphate lacking the base. Now what is happening? This gap is there now. So the DNA polymerase, then it comes into the picture. DNA polymerase will fill up the gap with the correct base, and then ligase will reseal the gap. This is how a base excision repair is taking place. Then the next type of repair we said is the nucleotide excision repair. Now the DNA that is damaged by UV light or through ionizing radiation or to other environmental factors, they often result in the modification of certain bases, strand breaks or cross linkages, etc. So the nucleotide excision repair will be ideally suited for such large-scale defect in the DNA. So if there is any defect again in the repair of this nucleotide excision repair does not take place, so it may occur in such rare conditions such as xeroderma pigmentosa. This is a condition, it is a rare autosomal recessive disorder which occurs due to a defect in the nucleotide excision repair of the damaged DNA. The affected patients will be photosensitive and susceptible to skin cancer. So here, this is an example with nucleotide excision repair. We are showing an example of in E. coli where a pyrimidine dimer, thymine dimer is formed over here. So here you can see here, this is the thymine dimer is formed over here, and the recognition of this abnormal pyrimidine dimer is taking place through the nuclease. So damage recognition is taking place and nuclease cleavage. And the cleavage of the nucleus will take place here. So here the strands will be unwound by the helicase and the endonuclease will cut on either side of the damage. So the endonuclease that will cut will be approximately about 20 to 30 nucleotide long. So on either side, the endonuclease will cut over here. So it will be around 20 to 30 nucleotide long. So it's not only the pyrimidine dimer, but more extension of the oligonucleotide will be cleaved by the endonuclease. Then again, this gap that is occurring as a result of the excised oligonucleotide will be replaced by DNA polymerase, and the sealing will take place by DNA ligase. Then mismatch repair occurs mainly during replication. So despite high accuracy that is taking place in replication and high accuracy, that the fidelity of replication is maintained by the DNA polymerase. In spite of that, defects do occur when the DNA is copied. So here, example where cytosine instead of thymine could be incorporated opposite to adenine. So opposite, always base pairing rule will always be maintained in DNA. So in adenine, the complementary base pair will be thymine, but this thymine may be replaced instead by cytosine. If cytosine is being replaced by thymine, what will happen? Mismatch repair. A mismatch occurs, and this mismatch repair corrects a single mismatch base pair. So here we are just showing here the exonucleotide proofreading of the DNA polymerase that is normally taking place. What will happen? DNA polymerase, as it goes on adding the new bases one by one, DNA polymerase, sometimes what will happen here, in the new strand here, instead of thymine, one cytosine is incorrectly added. Here you can see these are the adenine bases, and thymine has to be go on continuously be added over here. But instead of that, cytosine, one base is incorrectly added. What will happen is that it will block further elongation of the strand. So at that time, DNA polymerase, which also exhibit 3', 5' exonuclease activity, it will remove the unwanted base, and then DNA polymerase continue to add the correct bases. But in spite of that, still the we said that the DNA polymerase will collect the bases, but actually how this is occurring is a little bit more complicated, where what will happen, another enzyme is there, GATC endonuclease is there. Here we are talking about an example in E. coli, where adenine of each GATC is methylated. Now you can see this is a parental strand and this is a new strand where a mismatch base has mismatch base is present. Now this how will any enzyme recognize where the mismatch base pair is? So parental strand will always be methylated. So methylation will help in recognition of the parental strand, while new strand will not be methylated. So this GTA endonuclease, which is having several subunits, it will cleave on either side. This will identify the mismatch base, and cleavage will occur by exonuclease digestion, followed by exonuclease digestion of the defective strand and its removal. And here again this gap will be filled in by a bases through DNA polymerase, and sealing will occur by ligase. Now, if there is a defect in the mismatch repair mechanism, it may result in a condition that is known as hereditary non-polyposis colon of the colonic cancer. Then double-stranded breaks repair is there, where double-stranded breaks, they are dangerous, and we said it may lead to mutation, but it results in genetic recombination, which may lead to chromosomal translocation, broken chromosome, and finally cell death. So this double-stranded breaks, it may lead to genetic recombination, which lead to chromosomal translocation, broken chromosomes, and finally cell death. So several conditions may arise as a result of this double-stranded breaks. And this, how can this repair occur? This double-stranded break repairs can be repaired by homologous recombination or by non-homologous end joining. As we have seen in the beginning of the class, what is homologous recombination and non-homologous end joining? The same mechanism operates over here in case of double-stranded breaks repair. So homologous recombination also occurs in yeast, while in mammals, non-homologous end joining dominates. Here, so what happened in double-stranded break? Because if the accidental break occurs, loss of nucleotides due to degradation from the end is taking place. So either non-homologous end joining may take place, or homologous recombination repair may take place in double-stranded breaks repair. So if you just see, summarize the DNA repair mechanism, we said base excision repair. So a single base pair, it may be the defect will be as a result of chemical damage. The base will be removed by the N-glycosylase enzyme, and a new base will be added. While in nucleotide excision repair, please switch off your audios. Make it mute, please. All of you mute your audios. And then in nucleotide excision repair, what will happen? This also may occur as a result of chemical damage, and 30 base pairs or 20 to 30 base pairs will be removed, and then correct bases will be added. So every time, wherever in all the types of repair mechanism in the end, DNA polymerase will be filling up the gaps, and sealing will occur by DNA ligase. In mismatch repair, that occurs mainly as a result of replication, copying error of 1 to 5 bases may take place. So cutting of the strand by exonuclease digestion, strand cutting, exonuclease digestion, and repair by DNA polymerase and ligase. Double-stranded break repair as a result of free radicals and radiation, unwinding, alignment, and ligation, either homologous or non-homologous. recombination may take place. So what are the diseases that are associated with the DNA repair mechanisms? We already mentioned about xeroderma pigmentosa, that is seen in nucleotide excision repair mechanism. And other than that, you also have Cockney syndrome, where this also due to a defect in the nucleotide excision repair mechanism, where transcription factor 2H is defective, and these patients may present with stunted growth and mental retardation. While in xeroderma pigmentosa, what was the sensitivity to UV light and skin cancers may occur. Then, as a result of mismatch repair mechanism, the condition that may be seen is as a defective mismatch repair, hereditary polyposis colon cancer, or otherwise known as the Lynch syndrome, where defective gene is seen in chromosome 2, that is defect in MSH1 and 2 gene may be seen. Then another condition is there that is known as ataxia telangiectasia, where it is the sensitivity to UV light and lymphoreticular neoplasms may be seen. Then you have Fanconi's anemia, defect in the DNA cross-link repair is seen with the increased recurrence of cancer. Bloom's syndrome, where the defect is in the DNA ligase or helicase, and it results in lymphoreticular malignancies. But the most commonly what you have to remember is xeroderma pigmentosa, hereditary polyposis colon cancer, and the Fanconi's anemia. Then we have dealt with what is DNA recombination, what is the DNA defects that is taking place, and how the repair of the DNA damage is taking place. Now we will see what is do you mean by telomere and what is telomerase. We have seen that replication always takes place from 5' to 3' direction in the new strand. So this is the newly strand, this is the parental strand. Replication is taking place in the 5' to 3' direction in the new strand. Now what will happen is that the DNA polymerase, it is not able to synthesize the new strand at the 3' end of the new strand. So this is the at the 5' end of the new strand. So this is the 5' end, because in the lagging strand, the synthesis is taking place from 5' end to new prime. So it goes and landing in this direction. So this 3' end of the parental strand cannot be replicated. So this end of piece of the chromosome is called as telomere. The end of the parental strand where the new strand cannot be replicated is known as the telomere. So now our body has a mechanism to take care of this problem. So another enzyme, that is a telomere terminal transferase or telomerase enzyme, it takes up this job of replication of this end of the chromosome. So here this problem is taken care by the telomerase enzyme. So this telomerase enzyme, unless there is some mechanism to replicate telomeres, what will happen? The length of the chromosomes will go on reducing at each cell division. Suppose if this is not replicated, what will happen? Then this newly synthesized strand will become shorter and shorter. It will lead to gene loss, and the stability of the chromosome will be lost. So the shortening of the telomere end is prevented by the telomerase enzyme. What is this telomerase enzyme? It contains an essential RNA component. This telomerase enzyme contains an essential RNA component. This will provide a template for the telomerase repeat synthesis. So this telomerase will act like a reverse transcriptase. It will recognize the 3' end of the telomere. It will recognize this end, and the RNA template, RNA component of the telomerase will provide a template for synthesis of this small strand of DNA. So the small strand of DNA will be synthesized by the telomerase, and so RNA produced by telomerase is acting as a primer, and then DNA polymerase completes the synthesis of the lagging strand. So the importance of this telomerase enzyme is that it prevents the shortening of the chromosome. So I'll just show you a video. Do you have any doubts? Have you all understood?
Yes, ma'am.
What we said for the type of DNA damage that can occur? Single base damage alteration may take place. You may have double base alteration. You may have oxidation damage, or you may have double stranded breaks. And what are the types of repair that they may DNA damage may be corrected by repair mechanism? You have what are the repair mechanisms?
Base excision, base repair,
Base excision repair, nucleotide excision repair, mismatch repair, and double stranded break repair. Okay. And then there are some diseases that may be associated even if the DNA repair mechanism is defective. And we talked about the telomere and the telomerase, right? So now I'll be showing you one video. The video నేను పోస్ట్ చేసాను ఇది పోస్ట్ చేసావా ఇది ఓపెన్ చేసావా ఇది కూడా ఓపెన్ చేసావా ఇది వీడియో ఇది సారీ ఇది ఓకే నేను మై మిస్టేక్ అది ఓపెన్ చేసాను సౌండ్ మీరు మైక్ పెట్టడానికి సౌండ్ వస్తుంది
is initiated by NIX at the same position on the two parent. In the holiday model of homologous recombination, DNA exchange is initiated by NIX at the same position on the two parental DNA molecules. The nicked strands partially unwind, and each invades the other molecule by pairing with the complementary unbroken strand. Ligation of the broken strands produces a cross-strand intermediate known as a holiday junction. At this point, the DNA molecule need to be separated or resolved. Let's look at a simplified version of the DNA to see a model of how resolution can occur. In this model, the duplexes revolve around their connection point. Another revolution aligns the strands for cutting and resealing to form separate duplexes. The resulting two double-stranded molecules, which consist of exchanged segments, are now resolved.
So have you understood how the recombinant holiday junction is being formed?
Yes, ma'am.
Okay, so we will end the class now.